Welcome to the deep dive. Today, we're tackling something really revolutionary, bioinformatics.
Yeah, it's a big one.
We've got some fantastic resources here that really unpack the field. And our mission, well, it's simple. Give you the essential knowledge, the key insights, without getting totally bogged down in, jargon.
Exactly. Think of it as the digital side of biology.
The digital side of biology.
Yeah.
What it is, why it matters, and, frankly, the incredible things it's making possible.
Absolutely. At its heart, bioinformatics is the infrastructure and the smart tools we need to handle just enormous amounts of biological information.
We're talking massive scale, right?
Oh, yeah. The complete human genome sequence, countless medical images, mountains of data from drug discovery labs. It's just growing exponentially.
Wow.
And bioinformatics is, well, it's how we make sense of it all.
So it's like going from a few handwritten notes to this huge, ever-expanding digital library of biology.
That's a great analogy.
And to navigate that library, you need organization. That's where these biological databases come in. I see names like GenBank, EMBL, SwissProt.
You've hit on a key point.
Yeah.
Imagine if every scientist just kept their notes private. It wouldn't work.
No progress.
Right. So these databases, like GenBank for sequences or SwissProt for proteins, they're central public repositories.
Public, that's important.
Hugely important. They've basically enabled what you could call desktop research.
Desktop research.
Yeah. Scientists anywhere can access and analyze this incredible wealth of data without needing to be in the lab generating it initially. And they often specialize, one for genes, one for proteins. Makes finding things easier.
That makes perfect sense, like a specialized search engine for biology. And to frame all this data, the material reminds us of the central dogma. DNA to RNA to protein.
Yep, the fundamental flow.
And the different levels, too, from atoms right up to complex networks. It's all connected, isn't it?
Absolutely. That central dogma is key, and understanding those levels helps you appreciate how, say, a tiny change in DNA can ripple all the way through an organism. It gives us context.
Okay, good foundation. So with that laid out, let's talk evolution. Figuring out how different life forms are related. That's phylogenetic analysis. Yeah.
Precisely. Tracing the family tree of life using genetics.
How does that work, broadly?
Well, by comparing the genetic material, DNA or protein sequences, bioinformatics lets us reconstruct evolutionary history. The text mentions two main flavors, distance-based and character-based methods.
Okay, distance and character.
Think of it like this. Distance-based looks at the overall genetic distance or difference. Character-based focuses on specific shared features or traits. Did they inherit this specific mutation or not?
And when we visualize these relationships, we get these tree diagrams, phylograms, cladograms, phenograms. They sound similar.
They do, but they show different things. It's like different styles of family trees.
Oh, so
A phylogram, its branch lengths mean something, they're proportional to the amount of evolutionary change.
more change, longer branch.
Exactly. A cladogram focuses just on the branching order, who split off from whom. The lengths don't really matter.
Just the relationships.
Right. And a phenogram groups things based on overall similarity now, regardless of the exact evolutionary path. Like, who looks most alike today?
That's a really clear way to put it. Okay, moving on to mapping genomes. Finding where genes actually sit on chromosomes. Linkage groups, genetic crossover.
Yeah, so genes close together on a chromosome tend to be inherited together. That's a linkage group.
They stick together.
Mostly. But during meiosis, when sperm and egg cells form, chromosomes can swap segments. That's crossover.
Ah, so they can get separated.
They can. But the closer they are physically, the less likely a crossover happens between them. That helps us map their relative positions.
Makes sense. And the text distinguishes euchromatin from heterochromatin. Sounds like active versus less active DNA.
That's a good way to think about it. Euchromatin is more open, packed with genes being actively transcribed. Heterochromatin is tightly packed, lots of repetitive sequences.
And we know less about heterochromatin.
We know it's important for chromosome structure, things like that. But yeah, fewer genes, and its functions are still being figured out.
So how do we actually pinpoint gene locations? We see this alphabet soup of acronyms. RFLPs, VNTRs, STSs, ESTs.
Yeah, it can seem like that. Think of them as unique landmarks on the genome map.
Landmarks, okay.
Because they vary between individuals, they let us track specific DNA regions. RFLPs use enzyme-cutting site differences. VNTRs are variable numbers of short repeats.
And STSs and ESTs.
STSs are short, unique sequence tags, easy to find. ESTs are fragments of genes that are actually being expressed, turned into RNA.
Ah, so they mark active genes.
Exactly. By mapping all these landmarks, we build a genetic map. There's also physical mapping, which measures the actual DNA distance more directly.
Right, okay. Now, sequence alignment, this feels fundamental. Comparing DNA or protein sequences.
Absolutely fundamental. Pairwise for 2 sequences, multiple alignment for, well, more than 2.
And why do we do this?
It's like comparing texts to find similarities and differences. In biology, those similarities hint at evolutionary relationships, shared ancestry, maybe even shared function.
Because evolution involves changes like substitutions, insertions, deletions.
Precisely. Those events, indels as we call insertions, deletions, drive the differences we see. Alignment helps us spot them.
And this leads to terms like homologs, orthologs, paralogs, key concept.
Very key. Homologs are just any genes related by common ancestry. Easy enough.
Okay.
Now, orthologs are homologs in different species that came from a single ancestral gene. They often do the same job, just in different organisms.
Like the same gene in humans and mice.
Exactly. Paralogs are homologs within the same organism. They arose from a gene duplication event.
So copies within one species?
Right. And often, after duplication, one copy might evolve a new related function. It's a major source of evolutionary innovation.
Interesting. So when we do alignments, there are different approaches. Global versus local. Smith-Waterman, Needleman-Wunsch.
Right. Global alignment, like Needleman-Wunsch does, tries to match up the entire length of 2 sequences. Good for closely related
Well,
stuff.
you expect similarity throughout.
Yeah. Local alignment, using Smith-Waterman, is more about finding the best matching regions, even if the sequences overall are quite different.
Like finding a shared functional domain within 2 larger proteins.
Exactly that. Super useful.
And to score how good these alignments are, we use matrices like PAM and BLOSUM, based on real evolution.
That's the idea. They're built by looking at how often amino acids actually substitute for each other in known related proteins over time.
So a common substitution gets a better score than a rare one.
Precisely. It adds biological relevance to the alignment score. Helps tell us if the similarity is likely meaningful.
The text also mentions dot plots, simple visual tool.
Very simple, very intuitive.
Yeah.
Plot one sequence on the x-axis, one on the y. Put a dot where letters match. A diagonal line just jumps out at you if there's a similar region. Great first look.
Cool. Now, searching the massive databases, BLAST and FASTA are the big names here, right? The workhorses.
Definitely the workhorses. They're designed for speed, searching huge databases quickly for sequences similar to your query.
What's special about BLAST?
BLAST is particularly famous for its speed and, crucially, the statistics it provides. It gives you an E-value.
E-value.
Yeah, the expected value. It tells you how likely it is you'd find a match that good just by random chance in a database of that size. Lower E-value means more significant, more likely to be biologically real.
So it helps filter out noise.
Exactly. And it focuses on finding good local alignments, remember?
Yeah.
Oh, and it needs the databases to be specially formatted first. Lots of different BLAST databases available, too, for different sequence types.
And filtering low complexity regions first. Why do that?
Ah, right. Some sequences have lots of simple repeats, like A at A or at at at. These low complexity regions can sometimes cause lots of weak, meaningless hits in BLAST. Filtering them out cleans up the results.
Makes sense. Then there's PSI BLAST. Sounds like BLAST version 2.0, finding more distant relatives.
Sort of. Position-specific iterative BLAST. It does a normal BLAST search first.
Okay.
Then it takes the good hits, builds a profile or a consensus based on the conserved patterns in those hits, and searches again using that profile.
Ah, so it learns from the first round.
Exactly. Each iteration can refine the search and often pick up more distant evolutionary cousins that the initial simple search missed. It's much more sensitive.
Clever. Okay, last bit on alignment. Multiple sequence alignment. Clistal W, MFFT, dialign. Why align many sequences?
It lets you see patterns of conservation across a whole family of related proteins or genes. What's really crucial? What changes? What stays the same?
And different methods again. Progressive versus iterative.
Yeah. Progressive ones like Clistal W or MAFFT usually align the most similar pairs first, then progressively add the others. Iterative methods like dialign start with an alignment and then keep refining it to try and improve the score.
And UPGMA clustering, dendrograms.
UPGMA is a way some tools cluster the sequences based on similarity before aligning them to guide the process. And The result is often shown as a dendrogram, that tree-like diagram showing how the sequences relate within the alignment.
Okay, clear. Shifting gears now. Finding genes in genomes and how they're controlled starts with sequencing tech. Sanger versus next-gen, like Illumina, Solid, PacBio.
Yeah.
Huge changes there.
All massive. Sanger was groundbreaking, gave us the first genomes, but it was slow, expensive.
Right.
Next-gen sequencing just blew the doors open, way faster, way cheaper. Suddenly, sequencing loads of genomes became feasible. That flood of data is what bioinformatics has to deal with for gene prediction.
But just having the sequence doesn't automatically tell you where the genes are, especially in complex organisms.
Not at all. It's a real challenge. The text mentions two main computational strategies, ab initio and comparative.
What's the difference?
Ab initio methods try to find genes just by looking at the DNA sequence itself. They look for signals like start and stop codons, promoter patterns, characteristic sequence statistics.
Based on the raw code.
Right. Comparative methods, though, leverage what we already know. They compare the new genome sequence to known genes from other organisms.
So if a stretch looks like a known mouse gene,
It's probably the equivalent gene in your new sequence. Often gives clues about function, too. GNScan is mentioned as a tool using statistical models for ab initio prediction.
there's also exon trapping, an experimental method.
Yeah, a lab technique. It's a clever way to fish out exons, the coding parts of genes. You basically trick the cell's machinery into splicing your unknown DNA fragment into a known RNA if it contains an exon. Then you sequence what you trapped.
Neat trick. Okay, gene regulation. Turning genes on and off. Promoters, cat box, proximal elements, enhancers, silencers. These are the control switches.
Exactly. The switches and dials. Promoters are DNA regions near the gene start where the transcription machinery binds. The core promoter, with a data box, helps position everything correctly.
And the others fine-tune it?
Yep. Proximal elements are nearby regulators. Enhancers can ramp up expressions, sometimes from far away on the DNA. Silencers, well, they turn it down or off.
How do we find these computationally? De novo versus knowledge-based.
De novo methods try to find new regulatory motifs just by looking for patterns that are statistically overrepresented in regulatory regions. Like finding a sequence that pops up in your gene starts way more often than chance. Weeder is an example.
Discovering from scratch.
Right. Knowledge-based methods use what we already know. They use models, often position weight matrices or PWMs.
PWM.
Yeah, a PWM captures the preferred nucleotide at each position in a known binding site for, say, a specific transcription factor. Databases like JASPAR collect these PWMs. You scan the genome, looking for sequences that match these known patterns.
Okay, using prior knowledge. And finally here, gene regulatory networks, GRNs, the bigger picture.
Precisely. GRNs map out the complex web of interactions. Which genes regulate which other genes? How do they coordinate to control cell functions?
Like a circuit diagram for the cell?
Kind of. The text mentions they have nodes, genes, regulators, and edges, interactions, and they're often bipartite. Genes and regulators are distinct types, and directional regulators act on genes.
Okay, onto gene expression on a big scale. Microarrays. Looking at thousands of genes at once.
Yeah, DNA microarrays were a huge leap. They give you a snapshot of gene activity, which genes are switched on, how strongly in a specific sample.
How do they work?
They rely on complementary base pairing. You have a chip, like a slide, with tiny spots, each containing DNA from a known gene,
A spot for each gene.
essentially. Then you take RNA from your cells, convert it to labeled cDNA, that's the express stuff, and wash it over the chip.
And it sticks to its matching gene spot.
Exactly. The more label sticks to a spot, the more that gene was being expressed in your original sample. Great for comparing conditions like healthy versus diseased tissue or identifying genetic variations like SNPs.
But potential for errors, like any experiment. Tissue prep, labeling.
Oh, definitely. Lots of steps where variability can creep in. Sample handling, RNA quality, labeling efficiency, the hybridization itself. That's why good experimental design and data normalization are absolutely critical. You need statistics to clean it up.
Right. So once you have this massive data, how do you make sense of it? Clustering, PCA.
Essential analysis tools. Clustering methods, hierarchical, Cavanes, SOMs, group genes, or samples that behave similarly. Genes involved in the same pathway often have correlated expression patterns, so they cluster together.
Finding patterns.
Finding patterns. In PCA, principal component analysis is a way to reduce the dimensionality. It finds the main axis of variation in your huge dataset, making it easier to visualize and see the big trends.
The text also mentions tag-based methods, SAGE, TagSec, different from microarrays.
Yeah, different approach. Instead of hybridization, these methods involve sequencing short tags from the ends of the RNA molecules.
Direct.
Basically, yes, you sequence millions of these short tags. The number of times you sequence a tag corresponding to a specific gene gives you a direct quantitative measure of its expression level, often seen as more quantitative than early microarrays.
Okay. And CGH, microarrays. Different purpose again, copy number changes.
Right, comparative genomic hybridization. These look for deletions or duplications of chunks of chromosomes. Are there extra copies of this gene, or is it missing? Very important in cancer, where these copy number changes can drive tumor growth.
Got it. All right, let's dive into proteins. Structure and function prediction. The text outlines the hierarchy, primary, secondary, tertiary, quaternary structure, and mentions hydrophilic interactions driving folding.
Yep, understanding that hierarchy is key. Primary is just the amino acid sequence, the string of letters.
The blueprint.
Exactly. That dictates how it folds. Secondary structures are local patterns, like coils, alpha helix, and sheets, beta sheets. Tertiary is the overall 3D shape of one whole protein chain, often containing functional chunks called domains.
And quaternary.
That's when multiple protein chains team up to form a larger complex. And yeah, the way hydrophobic bits hide from water inside the protein while hydrophilic bits face out is a major driving force in getting that final 3D shape.
And we have databases for these structures, PDB.
The Protein Data Bank, PDB. It's the main global archive for experimentally determined 3D structures, mostly from X-ray crystallography and NMR spectroscopy.
What's in a PDB entry?
The atomic coordinates, basically the 3D map of the molecule, plus info on how the structure was determined, maybe some functional notes. It's grown enormously as the techniques improved.
And tools to see these structures, kinimages, ligplot.
Invaluable tools. You need to visualize these complex shapes. Kinimages give you interactive 3D views you can rotate and zoom. Ligplot is great for seeing exactly how a small molecule like a drug interacts with a protein which atoms are bonding, etc.
Now, predicting structure from just the sequence? The protein folding problem? Sounds hard. Leventhal's paradox.
It's the classic hard problem in biology. Leventhal's paradox just highlights the scale. A protein could theoretically fold in countless ways. If it tried them randomly, it would take longer than the universe has existed.
Well, it can't be random.
No, there must be folding pathways, guiding principles. But predicting that path computationally is tough.
So we have different methods. Secondary structure prediction, homology modeling, threading, ab initio.
A whole toolkit. Secondary structure prediction tries to guess the local helices and sheets. Homology modeling is powerful if you have a related protein whose structure is already known. You use that as a template.
Building on existing knowledge.
Exactly. Threading tries to see if your sequence fits well onto any known protein fold from a library of structures. Ab initio tries to predict it from scratch, using physics and chemistry principles. Much harder.
And specific tools for transmembrane proteins, like PhD-HTM, DS.
Yeah, predicting which parts of a protein span cell membranes is a specific important challenge, and those are tools designed for that job.
Okay, predicting function now. Even harder than structure sometimes, because context matters.
Often, yes. Function isn't just the sequence or structure. It's where the protein is, what it interacts with, the cell's state. It's complex. A protein can even have multiple jobs.
So how do we predict function computationally? Motifs, HMMs.
We look for clues in the sequence. Motif-based methods search for short, conserved sequence patterns known to be linked to certain functions. PROSIT is a big database of those.
Like little functional signatures?
Kind of. HMMs, hidden Markov models, are more sophisticated. They model an entire protein family, capturing the allowed variations. You can compare your new sequence to a library of HMMs to see which family it fits best, and infer a function from that.
HMMs sound complex, Viterbi, Bonewatch algorithms.
The maths can get complex, but the idea is using probability. An HMM has states, like match this position, insert here, delete here, and probabilities for moving between states, and for seeing specific amino acids in match states.
And the algorithms.
Viterbi finds the most likely way your sequence fits the model, alignment. Baumwelch is how you train the model, learning the probabilities from known examples.
Okay. And network methods. String, SMART, hydrophobicity analysis, more clues.
Exactly, more lines of evidence. String predicts protein interactions based on lots of different data types, gives functional context. SMART focuses on identifying known functional domains within your protein. Hydrophobicity plots can suggest if it's a membrane protein or has specific interaction surfaces.
Right, okay. Nearly there. Proteomics, studying all the proteins. More dynamic than the genome, you said.
Definitely. The genome is mostly static, the blueprint. The proteome is what's actually happening. If proteins are being made, how much are they modified? It changes constantly with conditions.
And proteomics has subfields: expression, structural, functional, proteogenomics.
Yeah, just different focuses. Expression proteomics quantifies protein levels. Structural proteomics wants to solve lots of structures. Functional proteomics investigates what proteins do, their interactions. Proteogenomics integrates genome and proteome data for a fuller picture.
Key tech: 2D gels, mass spectrometry, databases like PRIDE, tools like Mascot.
Absolutely central. 2D gels separate proteins by charge and size. You can see hundreds on a gel. Mass spectrometry then measures the mass of peptide fragments very accurately to identify the proteins.
Peptide mass fingerprinting.
That's one way mass spec identifies proteins. You pair the measured peptide masses to theoretical masses calculated from sequence databases. PRIDE stores the raw mass spec data. Mascot helps with that database matching.
And other tools: protein microarrays, affinity chromatography, yeast-2 hybrid.
Complementary approaches. Protein microarrays let you study many protein interactions or activities at once. Affinity chromatography uses specific binding to purify proteins. Yeast-2 hybrid and affinity purification plus mass spec, APMS, are key ways to find out which proteins physically interact in the cell. Lumiere is a fancier version for mammalian cells.
And finding gene families, post-translational modifications, PTMs. The story isn't just the sequence.
At all. PTMs adding phosphates, sugars, etc. can completely change a protein's function or interactions. Finding them is crucial. FindMod is a tool for that. And comparing proteomes across species helps identify gene families, orthologs, conserved functions.
Okay, final stretch: drug discovery. From history to pharmacogenomics, how our genes affect drug response. Real-world impact here.
Huge impact. We've moved from accidental discoveries towards rational design. Pharmacogenomics is tailoring medicine based on your genetic makeup. Why does a drug work great for one person but not another? Genetics often holds the key. The examples in cancer, asthma. They show the power of this.
And finding and validating drug targets, that first step.
Crucial. You need to find a molecule involved in the disease that you can safely target with a drug. The text mentions using genetics, expression data, then validating the target using things like RNA interference, RNAi, or antibodies to see if blocking it actually helps.
Then high-throughput screening, HTS, computer-aided drug design, CADD, using tech to sped things up.
Absolutely. HTS tests thousands, even millions of compounds automatically to find starting points. CADD uses computers to model drug target interactions, predict properties, even design new molecules digitally. Saves a lot of time and resources.
Like pharmacophore modeling, docking simulations.
Exactly. Pharmacophore modeling finds the essential 3D features a drug needs. Docking tries to predict exactly how a potential drug molecule fits into the target protein's binding site.
And predicting ADME tox properties, absorption, distribution, metabolism, excretion, toxicity, critical for safety.
Absolutely critical. A drug has to get to the right place, last long enough, not be toxic. Predicting these ADME tox properties early, often using computational models, helps weed out problem compounds sooner. Saves huge amounts of money and time. The text also lists key drug databases.
Final targets mentioned: GPCRs, ion channels, cell cycle stuff, biomarkers, hot areas.
Very hot. GPCRs are targets for a huge number of current drugs. Ion channels are tricky but important. Understand the cell cycle is key for cancer drugs. And biomarkers, measurable signs of disease or drug response, are vital throughout drug development and for personalized medicine.
Okay, last topic: pathways and systems biology, looking at the whole network.
Exactly. Moving beyond single genes or proteins to understand how they work together in pathways, metabolic, signaling. Systems biology tries to understand the whole complex system, its interactions, its dynamics.
Using databases like CatCore-G gene ontology.
Yep. CatCore-G maps out known pathways. Gene ontology provides a standard language to describe gene functions, which helps in analyzing these large networks. Tools like these help us organize the complexity.
And these networks have properties. Robustness, modularity, we can simulate them.
Right. Biological networks are often robust. They can handle some perturbations. They're modular, built from functional units. Metabolic control analysis, MCA, studies how control is distributed in pathways. And yes, building computational models in silico cells like E-cell lets us simulate and try to understand their behavior.
Wow, okay, that was truly a deep dive into bioinformatics, from the basic data right through to, well, drug design and systems thinking.
Absolutely.
Yeah.
And what really strikes you is how interconnected it all is, isn't it? Bioinformatics provides the glue, the tools, the framework to understand life's complexity at that molecular level.
And for you, our listener, we really hope this has given you some valuable insights, whether it's understanding science news, maybe for your career, or just plain curiosity. Grasping these ideas is, well, it's becoming pretty essential.
It really is. And, it makes you think, with data piling up even faster now, what are the next big bioinformatics innovations we're going to need? What new approaches will be critical for the next wave of discoveries and actually getting them into the real world? It's a really exciting frontier.